Hello students, welcome to you all to my new series of biology, and today I am going to cover the plant tissue culture of your syllabus. So first understand that what is plant tissue culture. So plant tissue culture is maintaining and growing the plant cells. That means it is a technique to maintain and grow new plant cell or new plants. So let's come to the literal meaning of the word plant tissue culture. So plant though is very common. You must be knowing that what is plant. So come to the word tissue. Tissue you all have studied in class 9 that what is tissue. Like tissue is what? Collenchyma, parenchyma, sclerenchyma, which is present in plants. So tissue, and culture means growing. Culture means to grow. So means we are taking tissue from the plant to grow a new plant or new plant cells from a plant tissue. Plant tissue either must be parenchyma or from the collenchyma. So actually plant tissue culture, what we are growing plant at home is in the soil or by the natural process. Plants are being excuse me. plants are being grown by the natural process in general, but this plant tissue culture is a new technique which is used to produce the plant in your required quantity or in the desired quantity or desired traits has been produced in this tissue culture techniques. Actually this technique is being used in the lab by the scientist. This technique is now this is not very new technique, but it is being practices from like 10 years, 12 years, 15 years or before that, but it is not so common. It is common but not so common. So come to this line, maintaining and growing the plant cells. So what we have to do, we have to take a plant. What we suppose we have taken a mango tree or not a banyan tree, a lychee tree or any of the tree. So we have taken a tree, we have taken a leaf from that tree, and from that leaf we have taken a and this tissue is called as explant. The part of the plant which we are taking to produce a new plant is called explant. So what we have taken an explant from the tree, from a leaf, that is the tissue. So we have taken tissue, like we will take a tissue in the petri plate or a dish. In common language, just understand, we have taken a plant tissue or a leaf tissue in a petri plates, and then we have given this an artificial medium. And what is that medium, artificial medium? Yes, the plants need what? Sunlight, water and minerals for the growth. But what we are doing, we are giving, it's a medium. Medium is what? Food for the plant. So we are giving an artificial medium to this explant. Explant means what? The part of the plant which is being growing. So we are giving some artificial medium to grow this explant into an artificial culture. So we have given and kept it under some environmental or the required temperature or required condition, whatever it wants. like in the lab there are some laminar flow or sanitization practices being done to grow this plant, and this explants after growing in artificial medium grow in number of plant, and this growing the small plants in a variety in many number of plant is called micropropagation. So a word also come here, micropropagation means small production of this explants from this small explants, means new plants is being grown in this medium. And what we are doing, like very we have taken like many bottles in the lab, and in that plant we have given the medium and we have put the explant, and that explant is being grown in many of the plants, and that growing of the many of the plants is called micro propagation. So this plant is the new plant which is being produced in lab is ready to being cultivated in the soil. So the benefit is that we have taken one explant, and from that one explant we have grown hundreds of plant in the artificial medium in the lab in the given medium, so that the plants has increased their variety, increased their species, increased their numbers in a good variety, in the good yield, and that plant is being ready to cultivated in the soil or in the field. So automatically the yield will be what? Increase. So coming to its benefit. So one of the benefits of this plant is what? It has increased the number. Suppose we have to grow a plant, so we can grow thousands of species in the lab very easily, and that plant will be grown in the soil or in the field. So number is being increased, yield is being increased. We can also increase the good variety, and one more very important benefit is that we can improve a good variety, means a healthy variety of a plant from the ill plant. That means if we have a tree, suppose this is a mango tree and this has some disease, this plant has some disease. So what we are taking from this plant is what? A tissue. So tissue is being taken from what? Either meristematic tissue is being taken. So either lateral tissue or apical meristem is being taken. So apical meristem grow very fast. So it has the ability to divide rapidly. It undergoes very rapid division. So after differentiation, what happened? The disease is being out from that explant, and disease variety is being gone out, and the healthy plant is being obtained from this explant. So this is the very important benefit of plant tissue culture. And one thing, it is given in a book also, that who has given this technique? Herbert Land. And the artificial medium which we need to give is what? Like we are giving some hormones, auxins, cytokinins, abscisic acid, and gibberellins. These are the hormones which is used as artificial medium for the plant tissue culture. So this is all about the plant tissue cultures you need to know. And one more thing, sorry, somatic hybrid or somatic hybridization. So what this is, what we have taken cell, plant, two cells, means cells of two different plants, So what we have done, we have crossed it. Like for this, suppose this is a plant cell and this is what? Cell wall, and this is what? Protoplasm. When the protoplasm is being crossed, what happened? A new variety after after crossing is being generated. Like in one plant, tomato and potato is being cultured to get tomato. You can say like this is what in animals also this is being done, cloning. Yes, so like that, this is what in animals hybrid species is there. So like plant, this is also being done. Not 100% this was successful, but it was successful. That's why, so a new variety, when tomato and potato was crossed, tomato was being given. So we have taken the protoplasm A and protoplasm B and crossed to get a new variety. So this was plant tissue culture.